Welcome to Lecture 12 Secret Codes. We're going to start off where we left off yesterday in our lab class, which is with this slide, thinking about what the structure of DNA is. So we did a puzzle and we were looking at the structure of the DNA. Let me review some of the vocabulary. The first part of our DNA is the sugar. So DNA is made out of the sugars, which is the little pentagon-shaped thing with an S in it. The name of the sugar is called deoxyribose. It's a type of simple sugar. Remember, simple sugars are things like glucose or galactose, things that are simple sugars that are not bound to anything else. But these sugars are bound to something else. They're bound to a phosphate, which is the little P-shaped molecule. So sugars and phosphates bound together in kind of an alternating pattern make the sugar-phosphate backbone of the DNA. One on each side, kind of like the arms of the ladder. In between, we have what are called bases, and bases are the third component of DNA. There's 4 different varieties of bases. There's adenine, thymine, guanine, and cytosine. And adenine and thymine are bound together, whereas guanine and cytosine pair up together. So you have adenine and thymine pairs, or guanine and cytosine pairs. And the unique thing about the bases is that any base at all could be attached to a sugar in the middle of the ladder. So you can have an A, a G, a T, or a C at any spot. And that's what makes DNA a really useful code. So what happens is that you have this DNA molecule, and you have these A's, G's, C's, and T's kind of arranged in the middle. They pair up together down the middle. And that is called the base pair. In a moment, I will show you how to read the base pairs and use those base pairs to actually make an organism. Another few vocabulary words you want to know is called, one of them is nucleotide. A nucleotide is a phosphate, a sugar, and a base. So for instance, if you look in the upper corner, a P, an S, and an A, those 3 components together is a nucleotide. And what happens is when our digestive system digests DNA, it will break DNA down. For instance, that DNA we found in the strawberry, it will break it down into a nucleotide, and then we can absorb it into our bodies. And then our bodies can use those nucleotides to make more DNA. So for instance, you could take that P, the S, and the A, that nucleotide, and we could break down the strawberry's DNA, and then if our cells are trying to make more DNA, like a cell is trying to make a new red blood cell, then that nucleotide can be part of our DNA, which also says that every DNA is the same. So all of the DNA that we find in living things, with some exceptions, very few exceptions, but most of the DNA, especially the ones that we eat, are ones that we can use and break down into nucleotides, and then use those nucleotides to build up our own DNA when we need to make more. A couple other things that we need to know are the differences between covalent bonds and hydrogen bonds. Covalent bonds are, in the picture, drawn as a solid line. And a covalent bond are the kinds of bonds that we're most used to talking about. They are bonds between atoms where they're sharing electrons. And covalent bonds are incredibly strong. It's very difficult to separate 2 atoms that are sharing electrons. They really like to share those electrons, and therefore it's very difficult to break them. They are very strong bonds. So what that means is that, for instance, if you look at the backbones, or if you look at the connection between a sugar and a base, all of those are solid covalent bonds, and those are very strong, which means that those backbones really like to stay together, which is why DNA as a strand, you can actually lift up the whole molecule with that bamboo skewer. It's a very strong molecule. It has a tendency not to break, which is a very good thing. It's a molecule that likes to stay together. Those backbones are incredibly strong and they won't break apart. However, we have hydrogen bonds, and hydrogen bonds are represented by the dotted lines. And the hydrogen bonds connect the base pairs together. And what's special about a hydrogen bond is that they aren't sharing electrons. Instead, what happens is that if an atom shares an electron somewhat unequally, so for instance, if you have a water molecule, the oxygen keeps the electron a little more than the hydrogen keeps the electron. And because electrons are negatively charged, that makes the oxygen slightly negative, and it makes the hydrogen slightly positive. And if you have water molecules near each other, the oxygens are slightly attracted to the hydrogens of a different water molecule. And that's because if something's a little negative and something's a little positive, those two things are attracted together. And it's very much the same way that a magnet works. So you can think of hydrogen bonds more like a magnetic attraction, whereas covalent bonds are like glue. You've glued them together versus you've got a magnet holding it together. So the hydrogen bonds are what's connecting the base pairs. The base pairs are not sharing electrons. Instead, the A is slightly negative and the T is slightly positive, and so therefore those two things are attracted together. You might notice that the base pairs, the A's and the T's, have 2 little dotted lines, and the G's and the C's have 3 little dotted lines, and that's because the A's and the T's have 2 hydrogen bonds. So that means that there's 2 places where there's a magnetic attraction. Whereas the G's and the C's, there's 3 places where there's a magnetic attraction. So the G's and the C's are slightly more magnetically attracted to one another through those hydrogen bonds than the A's and the T's. And what that allows you to do is it allows you to split the DNA apart down the middle like almost like Velcro, like you're ripping the Velcro apart, ripping one backbone apart from the other backbone. And it allows you to have some really unique things about the DNA, which you will see over in this lecture and also the upcoming one. So I think that covers all of our vocabulary. DNA obviously is the whole molecule, and it's the genetic material of most living things. Now, how do you read the DNA? How do you get the DNA to turn into something? So first, to kind of begin thinking about codes, I'd like you to turn to page 77 in your lab manual. It's called Secret Codes, and what it has is a Morse code message. So hopefully you have found the page and you are noticing that you can take the Morse code, and the Morse code is a series of dots and dashes. And if you find 2 little dots, that would represent an I, which is the first thing in the secret message. And then you need to find something that's a dot, dash, dot, dot. And when you find a dot, dash, dot, dot, you can match that up with the Morse code, and you can find the second letter in the secret message. So I would urge you to pause the presentation at this point and then start it back up when you are ready for the next part, when you've decoded the message.